going to start learning about the autosomal linkage. First of all, we need to understand what is the meaning of linkage. Now, if you guys remember, I told you guys in the beginning of this chapter that when the Mendel performed his experiments, he said that there are no chromosomes, there are only factors, elementins are there. But later on, with the discovery of chromosomes and with the help of other experiments, scientists came to know with one thing, that let's suppose if this is one chromosome, and on this one chromosome there is not only one gene. There can be many genes present on one chromosome. Let's suppose that on this chromosome there is A, B, C, D, E, F, G, H, I, J. These are the genes present on the same chromosome. Now, where the A will go, the J will also go there. Because they are riding the same train, right? They are riding the same car. So wherever the B will go, G will go there. It means that they are linked with each other. So this phenomenon is called linkage. Now first of all, I am going to tell you how it was discovered, right? And then we'll take different examples to talk about it. Now, there was a scientist, his name was Thomas Hunt Morgan. Now, in 1902, when the Mendel's work was discovered, when the Mendel did his work, no one paid attention to his work. They said that he's a mad person. We don't know what he's talking about, right? He died. But in 1903, that there were two scientists who were going through the books and they found his work and they analyzed his work and they said this is a groundbreaking ideas, right? And they started working on the plants. But Thomas Hunt Morgan and his associate and his team of workers and students, they were the first scientists who started working on animals. And they worked on Drosophila melanogaster. Now, this is the biological name, binomial name of the fruit fly. Now, this is a small fly which specially goes to the fruits, right? Collects the nectars from there. Now, why did they take this Drosophila melanogaster, the fruit fly? Always choosing an organism for the experiment, there can be many reasons behind it. One of the reasons should be that the life cycle should be short. Now, what is the benefit of having short life cycle? In short period of time, we can repeat the experiment very quickly, right? Second, that the progeny produced should be greater in number. Now, if the progeny produced or the offsprings produced are greater in number, we get less chances of error over there and we can easily find the ratios which will be generated over there. Now, when the Thomas Hunt Morgan, he started studying, first he studied the monohybrid inheritance of Drosophila melanogaster, all the rules which the Mendel explained about the segregation of the alleles, they were correct. They became very happy that okay, this is fine. After completing that, they said let's go and do a dihybrid cross, not this time with the plants, this time with the animals, and let's take the Drosophila melanogaster. Now, for the first time, they took two traits. They chose the long wing and vestigial wing. Now, this wing shape and wing type, this is one trait. This is one trait. Here we are talking about wings, long vestigial. Do you know what is vestigial means? We have discussed this thing before as well. An organ present in your body, but it is useless. Like? Right? Like appendix. That's what I was saying. You thought wrong. So long wing is the wing which helps in flying, but vestigial wing is a smaller in size, does not help in flying, but it is there, right? Second trait they observed, that was broad abdomen and narrow abdomen. Now, they already have find out before that long wing and broad abdomen, these both are dominant traits. And vestigial wing and narrow body, these both are recessive traits. Now, how did they come to know? If you take a long wing and cross it with the vestigial wing, all the progeny of the F1 generation will be what? Long wing. It means that long wing is a dominant trait. Got it? Same thing, you take broad abdomen and cross it with the narrow abdomen fly. All the F1 offsprings, they are broad abdomen. It means that it is what? A dominant trait. We can prove this over here as well. What did he do? Now remember that this is P1. P1 means what? First parental generation. P1 means first parental generation. He took two breeds of long wing and broad abdomen, vestigial wing and narrow abdomen. He crossed them with each other, and in result he got all the organisms of the F1 generation. They were long wing and broad abdomen. Even this experiment proves what? That these both traits are what? Dominant, and these both traits are what? Recessive. Now, what do we do always do? We always cross. Now, this is what? This is F1. Right? Now, we always do, we cross F1 with F1. What did he do? He took the long wing and broad abdomen. Long wing and broad abdomen. Now, he had the idea in his mind that whenever you cross over dihybrid inheritance of F1 and F1, you get what ratio? 9:3:3:1 ratio. And there is always production of what? Two new phenotypes. That is what he was expecting. That is what he was expecting. But what was the result he got? In this cross of F1, F1, instead of 4 phenotypes, he just got 2 phenotypes. What were those 2 phenotypes? They were long wing, broad abdomen. They were vestigial wing and narrow abdomen. Only these 2 phenotypes, and moreover that the ratio was what? 3:1. Now, this is a dihybrid inheritance, but the result, the ratio that we are getting, that is of what? Monohybrid. Only in the monohybrid we learned before the ratio comes 3:1. But this is a dihybrid inheritance. We are studying two traits at one time. Now, why we have 3:1? They said that maybe we have done something wrong in the experiment. They repeated the experiment. Again, same result, 3:1. Again repeated the experiment, again 3:1. The whole team sat down. They said, no, there is nothing wrong with our experiment. There is some other forces in play. Sorry?
Number of traits.
Same chromosome. Then they came to the idea that these traits, long wing and broad abdomen, they are present on the same gene, same chromosome. Now, as they are present on the same chromosome, they are not assorting independently. Remember that I taught you law of independent assortment, right? The Mendel said that during the gamete formation, the alleles, they assort independently. They are not attached with anything. But here, his law of independent assortment is what? Rejected, right? But it is applicable. It can be applicable if the genes are present on different chromosomes. If the genes are present on different, remember, I told you, Mendel was lucky that he chose 7 traits, and all 7 traits, they were on different 7 chromosomes, right? So here, and at that time, you see, I told you that if he would have chosen the 8th trait, all of his findings of 9:3:1 ratio will have gone -0. So here, and you see, remember, he was the only single person who was working on the genetics at that time. Here it is a team of geneticists, right? So alone, he could have rejected his own work, but here, they put their mind to it and eventually they came up with the solution, right? And what is the solution? That on same chromosome, 2 genes are present of 2 different traits, and they do not assort independently, they move together. Now, till now we have learned one thing. What? If there is a dominant trait, just write the capital letter. If there is a recessive trait, just write the small letter. But here, in this dihybrid cross, and even in the dihybrid cross of the plants, of the pea plant for tall and the yellow, whatever we were doing, right? What we were doing? We were writing simple letters, capital D, capital D, capital Y, capital Y. Is it possible to write in that way here? No. We cannot represent here that in this way. So here, the representation of the alleles or genes is not a simple method. Here is represented as chromosomes. Now I am going to show you guys how we represent it. Now, first of all, what do we have? The P1 type, we had long wing and broad abdomen, and we had to cross it with vestigial wing and narrow abdomen.
So what year was this studied?
Sorry?
What year?
1926. 1926, these experiments were going on. Alright? In the 1920s. Now, how do we represent? We make 2 chromosomes. Right? Let's suppose that we take capital L, right, for the long body. And remember, I told you this is what? True breed, pure breed. And for the broad abdomen, what do? We take capital B, capital B. Right? Now, for the vestigial wing, we can take small l. We can take small l, but we are taking VG. Right? VG. And this is what? Small b, small b. Understand, everyone? So this is what? Now this is the, over here, this is what? This is the phenotype. And this is what? This is the genotype. Next step, what do? We write the gametes. Now, how we will write the gametes? Simple. Capital L, capital B, circle. Right? Now, if I will write the gametes for this and this, will it be same or not? So I am writing it only once. Same over here. It will be what? VGB, circle, gamete. Yes.
Why do we use BG and not N?
Let's use L, small l. Don't be confused, right?
Yes, okay.
Small l, small l. Small l. Actually, at that time, the scientists used VG. I was following that thing, right? That old procedure. Now, what will happen? They both will join together, and the new organism that will be formed, that will be capital L, capital B. Small l, small b, and its phenotype will be what? Long wing and broad abdomen. And this is what? This is F1. What is this? F1. Now what do? We always cross F1 with F1. Right? Now, again, what do we have? Long wing and broad abdomen. Long wing and broad abdomen. And again, this is what? This is the phenotype. Now what I am going to do? I am going to write genotype. Now genotype for both of them will be what? This one. Capital L, capital B. Small l, small b. Capital L, capital B. Small l, small b. Right? Now what we'll do, we'll make gametes. The gametes over here, one of the gametes will be capital L, capital B. Other one will be what? Small l, small b. Same over here, capital L, capital B. And the other one will be small l, small b. Now what we can do? We can simply make a Punnett square over here. Now, let me make the Punnett square a bit so that you can find. I am making the Punnett square over here. Now, why I am using these lines? I am representing chromosomes actually. These lines are representing what? Chromosomes. Because this is a linkage. Because this is what? Linkage. Understand? This is a special kind of inheritance. So we cannot simply take capital L, small l. Understand? We are using these lines to represent what? Chromosomes. I will explain this again.
Yes. Small capital L small crossover.
Wait, we are not finished yet. This is just beginning of the today's topic. Alright? So guys, let's suppose here, over here we have male, over here we have female, right? Now, let's suppose that this is capital L, capital B. We circle this. This is small l, small b. We circle this. Right? Over here as well we have what? Capital L, capital B. We circle this. We have small l, small b. We circle this. Right? Now, what happens? This and this join together. It makes what? Capital L, capital B. Capital L, capital B. So what it will be? long wing, broad abdomen. I'm writing LB like before, but you will write complete. This one joined with this, so it will be what? Capital L, capital B, small l, small b. Again it will be long wings, broad abdomen. Got it? This one joins with this one, so it will be what? Capital L, capital B, small l, small b. And it will be also what? Long wing, broad abdomen. But when this one will join with this one, it will become what? Small l, small b. Small l, small b. So it will be what? Vestigial wing and narrow abdomen. Right? So in this case, what we are getting, I am mentioning over here, right? Because I want to save this space for the next question which he asked right now. That is the continuation of this. Now, here we got two phenotypes again, which are actually the parental phenotypes. We got long wing and broad abdomen. Then we have what? We have vestigial wing and narrow abdomen. Now, if you count 1, 2, 3, 3 are long wing with broad abdomen, and 1 is narrow body and vestigial wing. Alright? So, this is what? This is the simple inheritance. This is the linkage inheritance. Now, we can perform another experiment. We can perform this test cross on this one as well. Now, let's suppose, you see, we got three of these, right? Now remember that when we performed the test cross and it was a heterozygous condition, what was the rule that we made? Who remembers? If it was a heterozygous, remember, what did we learn? That there is a tall plant. There are two chances. This tall plant can be capital T small t, or it can be capital T capital T. Do you remember this or not?
So sir, if there is a short plant, then it is
It is always small t small t.
Cross it with the recessive.
But remember, we make the test cross only for the dominant trait to find out its genotype. Now, the tall plants are of two categories, right? Either they can be capital T small t genotype or capital T. Now, what was the first rule? Who remembers?
cross it with a short plant. You cross it with a short plant.
No, obviously, but what was the first rule that we made out of it?
Sir, half of the offspring will be dominant.
No, what?
Listen, let me remind you now, you guys did not study at home. Now, let me remind you, if the organism under observation is homozygous, all the offsprings of the test cross will be dominant. Understand? And if the organism under observation is what? Heterozygous, then 50% will be dominant, 50% will be recessive. Right? Now, again, we have these three. Long wing, broad abdomen. We have long wing, broad abdomen. We do not know which out of these three, which one is homozygous and which one is heterozygous. If we want to find out the test cross, remember, over here it was monohybrid. This is what? Dihybrid. This is what? Dihybrid. Now, another proof that this is autosomal linkage, if we perform the test cross of the dihybrid of autosomal linkage, the result will always be 1:1. I am going to perform that for you guys, right? Then we'll move further. Example does not finish here. But first we are going to do the test cross. Now, please pay attention. On one side, we have what? this long wing, broad abdomen. You know that in the test cross we always cross it with what? With the recessive parent, right? So this is the, over here we have recessive P1. And what is that? That is the vestigial wing and narrow abdomen. Now please pay attention. Let's suppose that over here, this one is what? This one is capital L, capital B, small l, small b. And this one you already know. What is this? Small l, small b, small l, small b. Right? Now the gametes formed over here will be capital L, capital B, gamete. Small l, small b, the gamete. Over here the gametes will be what? Small l, small b, small l, small b. Alright? Now what do? We make their Punnett scale. Here, let's suppose we have male, here we have female. Let me use a different color. Let's suppose this is capital L, capital B, circle. Here is small l, small b, circle. Here it is what? Small l, small b. Small l, small b. Right? Now, what happens? This one joins with this one, it will become what? Capital L, capital B, small l, small b, it will be what? Long wing and broad abdomen. This one joins with this one, it will become small l, small b, small l, small b, it will be vestigial wing, narrow body. Right? This one joins with this one, it will become what? Capital L, capital B, small l, small b. It will be what? Long wing, broad abdomen. This one joins with this one, it will become what? Small l, small b. Small l, small b, it will become vestigial wing, narrow body. Right? Now what do? Now we have two phenotypes which we got. It is what? Long wing and broad abdomen. And we have vestigial wing and narrow abdomen. Now there are two of these and two of these. We always simplify. It will 1:1. Now, during the test cross, if you guys please pay attention, very important thing I'm going to talk about. When we did test cross for this, we did the two test crosses, right? For the heterozygous as well and for the homozygous as well. I did not do here for the homozygous because from that knowledge we already have experience. If it is homozygous, dominant, and it is crossed with what? Recessive parent, the result will be what? All dominant. All dominant. Here, that is why I only took the example of what? Where it might be what? Heterozygous. Where it might be what? Heterozygous. So I took the heterozygous and I got the ratio 1:1. But the thing is that this is a dihybrid cross. This is a dihybrid cross and showing the inheritance like a monohybrid. Showing the inheritance patterns of monohybrid. So this is another proof. This 1:1 is another proof that the genes, they are what? Linked together. They are present on the same chromosome. Understood? Let me repeat this. We have another step ahead of this. But let me start again from here and go back there so that we can continue. Thomas Hunt Morgan wanted to prove that if we do dihybrid cross on an animal, we will get a 9331 ratio. For that purpose, he took Drosophila melanogaster, the fruit fly, and he took two contrasting traits, which are totally different from each other. He took long wing, he took wings and abdomen. There were two types of wings, long and vestigial, broad abdomen and narrow abdomen. With his previous experiments, he already knew long wing is a dominant trait and broad abdomen is also a dominant trait. Vestigial wing is a recessive trait and narrow abdomen is also a recessive trait. When he crossed them together in F1 generation, he got all the what? Long wing and broad abdomen. Then what did he do? He crossed F1 with F1, thinking he will get 9331 ratio. But interestingly, he got what? 3:1. And he got only the parental types. Long wing, broad abdomen and vestigial wing, narrow abdomen. I told you guys that they repeated the experiment many times, but still the same result came. Then they came to know that there is nothing wrong with their experiment. There is some other thing. Then they came up with the solution that these genes are not present on different chromosomes, they are present on the same chromosome. As the genes are present on the same chromosome, it is called what? Linkage. And they represent the linkage in this way, that we draw a line which represents a chromosome. The line drawn represents a chromosome. Understand? And we write the alleles like this. Right? Now, this is a true breed, this is a true breed. When we make the gametes and they join together, we make long wing, broad abdomen, that is F1 generation. And when we cross F1 with the F1, these both alleles, they always part together. They always move together. They do not separate because they are close to each other on the chromosome. So on these four types of gametes are formed. When we make the Punnett scale, we get only 3:1. Understood everyone? To further prove that these are what? These are linked together genes, we perform the test cross. And in the test cross, what do? That we take the example of a heterozygous organism, not the homozygous, because we know, according to our previous knowledge, if we take the homozygous, result will be what? All the offsprings will be dominant. So we took what? Heterozygous one, crossed it with the recessive parent.
Do we need that?
No. Right? So he crossed it with the recessive parent. Now, these gametes are formed, and at the end of the Punnett scale, what did it become? We got the 1:1. Now, even though it is a dihybrid inheritance, but the results are like monohybrid. So this proves that the genes are linked together. They are joined together. A question should have come into your mind. Sir, crossing over can take place or not? Crossing over can take place or not? If the crossing over takes place, then what happens we are going to discuss now. Alright? Now I am going to rub this from here. You can take the screenshot from the video. Right? I'm not stopping the video. Okay. Now I am rubbing from here. Now, if the crossing over happens, then what will happen, right? Now, look here, boys. Now, for crossing over to observe, we cannot take this, we cannot take this. There will be no result of the crossing over there because the alleles are what? Exactly the same, homozygous. For observing the crossing over, we have to take capital L, capital B, small l, small b. Right? Now, to show the crossing over, let's suppose crossing over happens like this.
So does it always happen or sometimes?
Remember, we have learned crossing over can happen at different places. At different places. Now, the crossing over was not taking place, that's why they got this result. But if the crossing over was taking place, then the other result came. Right? Now, what happened? Alright, boys. Please pay attention. Now, 4 types of gametes will be formed over here. 4 types of gametes will be formed over here. One type of the gamete probability is what? Capital L, capital B. Another probability is what? Small l, small b. Obvious or not? The parental type, the original type. Another, capital L coming and joining with what? Small b. So another can be capital L with small b. Alright? And small l, capital B. Understood everyone? Now, if there is cross of this with, let's suppose, a homozygous recessive parent. If a cross of this organism is with homozygous recessive parent, now let me draw the homozygous recessive parent. Now instead of writing, now let me write the both genes over here, sorry, both chromosomes over here first. It can be small l, small b, small l, small b. Now instead of writing all of them, I am writing what? One small, one b, this is one gamete. Right? So this is what? This is P1 recessive. It's gamete, right? Now please pay attention. If this one binds with this one, what it will form? Capital L, capital B, small l, small b. What will be the phenotype over here? Long wing and broad abdomen. If this crosses with this one, it will be what? Small l, small b. Small l, small b. What it will be? It will be vestigial wing and narrow abdomen. Understood everyone? Next step, Abdullah. If this one joins with this one, then it will be what? Capital L, small b, small l, capital B. Sorry, both are small. Now, a new phenotype. This will be what? Long wing and narrow abdomen. Long wing and narrow abdomen. And if this joins with this one, so it becomes what? Small l, small b, small l, capital B. Now this time it will be what? Vestigial wing and broad abdomen. And this is 1 ratio 1 ratio 1 ratio 1. Still it is what? 1 ratio 1 ratio 1. Yes. Because of linkage. Because of linkage. 9331 ratio only comes if the chromosomes, these genes, they are present on separate chromosomes. They are present on separate chromosomes, or I said another thing. Or far apart means that when I drew the diagram of a chromosome today, I said A, B, C, D, E, F, G, H, I, J. Now A and J, they are far apart from each other, right? During crossing over, they can assort independently. Understand? So remember one thing, independent assortment only takes place if the chromosomes are present far apart from, sorry, the genes are present on the chromosome are far apart, or genes are present on separate chromosomes. Alright? So this is one example of linkage. There are more examples as well. We have time, I am going to give you guys another example to solve it. Right now, right? Just now, okay.